Welcome to the cutting edge of cardiovascular sciences. I'm your host, John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist Research Institute and Houston Methodist Hospital. Joining me in the studio is Dr. Palo, who's a research associate in our Department of Cardiovascular Sciences. And today we have a very special guest, Dr. Ferid Murad. Dr. Ferid Murad is co-winner of the 1998 Nobel Prize in Physiology or Medicine for his paradigm-changing discovery that nitric oxide, a gas, is a signaling molecule for the cardiovascular system and relaxes blood vessels. We now know that this simple molecule, nitric oxide, again a gas that is released from cells, can act in other ways too. It's a neurotransmitter in the brain. It acts as a self-defense against pathogens such as SARS-CoV-2 and other viruses and bacteria. It also keeps our vessels open and free of disease. It can affect our metabolism and our physical performance. In addition to winning the Nobel Prize, Dr. Murad is the recipient of many awards and honors, including the Robert Lasker Award for Basic Medical Research, the American Heart Association SIBA Award, and he is a member of the National Academy of Sciences. Thank you, Dr. Murad, for joining us on the show.
Thank you for having me, John.
Well, you've had a remarkable career, and we're going to get in a moment to the work that you've done with nitric oxide and its role in cardiovascular health and disease. But I thought maybe we could start by talking to you a little bit about your early life and your interest in science and what sent you down this road? What sent you down this road toward the Nobel Prize? Tell us a little bit about your past, your background.
I grew up in a small community, Whiting, Indiana, in the northwest corner of Indiana. Between Gary and Chicago, we were about 15 miles from downtown Chicago. I went to a public school in Whiting. Fortunately, they really had some excellent teachers, and I was very influenced by many of the teachers I had in biology and chemistry, physics, mathematics, and many became friends. And some of them would come to the restaurant that my parents owned, and I would have lunch or dinner with them in our restaurant. And as I thought about college, what was I going to do? I was probably the second student on both sides of the family to ever finish high school and the first to go to college. And my parents couldn't afford to pay for my college education. But somehow I and my two brothers all got college degrees, two younger brothers. One became a dentist, one became a PhD anthropologist and professor at Chico State in California.
That's a rags to riches story with your father being an immigrant from Albania. So I think that's a pretty classic for American tale, isn't it?
Very, very common. You know, there are many. He immigrated to the U.S. in the early 1900s, probably around 1913, 1917. And when he came through, he had an Arabic name, an Albanian name, Ferid Murad Ayupe. And he met with he was selling candy on the street corners as a youngster to survive, and met some young folks from Austria who were coming to the U.S. through Ellis Island, and he joined them. And when he got to Ellis Island, the immigration agent asked his name, and he told him, and he said, "No, you're John Murad," and stamped his papers. So most of the immigrants had their names changed.
It made it easier for me to introduce you.
And what is very, very common with most immigrants, I'm sure you've seen this with people from Ireland and Poland and all over the European countries, they would go and visit with people they knew, some of their family members who were in the States or some of their friends who had immigrated previously. So when my father came through New York, he decided to go to Cleveland. He had a friend in Cleveland. He worked in the steel mills for a while. And then he went from Cleveland to Detroit and worked in steel mills there as well. And then decided for some reason to go to Chicago. He had a good friend in Chicago. And he was raising and taking care of horses on the North Shore of Chicago, near Lake Forest, where I lived for a while, and was a landscaper taking care of their lawns. And then decided, as many Albanians do, get into the restaurant business. So he had a restaurant in Chicago, And then he decided to create a restaurant in Whiting and moved to Whiting right across the border of Illinois and Indiana. And my mother was from Alton, Illinois, across the river from St. Louis. And she one summer went to work in her aunt and uncle's restaurant. The aunt had married a Turkish fellow, and on Saturday nights they would have coffee breaks and play cards, Turkish coffee and cards. It was very, very common with the Albanians and the Turks and many of the Macedonians. And my mother went to work there that summer, and that's how she met my father. And apparently they fell in love. She returned to Alton, Illinois at the end of the summer, and in August my father called her sister, went to Alton, extracted her from the house, put her on a bus and kidnapped her and took her back to Indiana and married her. She was only 17 years old.
Seems like both your parents were risk takers, a bit like yourself.
And that's how we grew up in this restaurant. And we worked, my brothers and I would wash dishes, wait tables, whatever. And the question was, how could we afford to go to college? My parents couldn't help us. And I looked around and got advice from various teachers. I took the examinations to go to one of the academies and decided not to do that, because if you go to a Navy academy or one of the military academies, it's mostly mathematics and engineering or business training, not biology and chemistry, not pre-med. And if you go to an academy, you have to pay back 4 years in the military when you finish the training. And then I would have to go back to college for a year or two to get more chemistry and biology that I wouldn't get to the academy, so I decided not to do that. But instead, one of my teachers had graduated from DePauw University, a small liberal arts school about 40 miles west of Indianapolis. And there was a scholarship program, and he took me down. I took the exams. I was able to get a scholarship for my tuition. And by teaching laboratories, anatomy, embryology labs, waiting tables, working 1 or 2 jobs each summer, I was able to get through college without any debt, which was very helpful. And it was there that I met my wife, who was a classmate at DePauw. And right after college, we got married. And I decided to go to Washington University because she was from the St. Louis area. And I thought as a medical student, it'd be nice for her to have family as I was busy in school. But then in January of 1958, I learned from a fraternity brother and one of my advisors, Forrest Fuller, who was an anatomy teacher, that there was a new program starting in Cleveland at Western Reserve University, the MD-PhD program. It was the first school in the country to start the MD-PhD program, which we know now is the MSTP program, the Medical Sciences Training Program. And I was the first student in the program. And I was so impressed during my interviews, I decided to do that. And after my interview in Cleveland, I returned to school and I said, Carole, we're not going to Washington University in St. Louis, we're going to Cleveland. And it's not 4 years of medical school, it's 7 years for both degrees, MD and PhD. That's what I did.
And that was a great decision because it gave you a chance to work with Earl Sutherland, Nobel Laureate for
my
cyclic AMP.
advisor was Earl Sutherland and Ted Rall. They were a team working with cyclic AMP. In 1957, they were interested in the effects of hormones on glycogenolysis, the degradation of glycogen by phosphorylase. And they found that working with liver extracts, they produced a new molecule, an intracellular messenger, that regulated phosphorylase activity to produce glucose from glycogen. They worked with epinephrine and glucagon. And it was known then from the work of Alquist, who was down in, I believe he was from Atlanta, that there were 2 classes of adrenergic receptors, alpha receptors and beta receptors. And Sutherland said, Murad, tell us which receptor is being influenced by epinephrine. Is it alpha or beta? And that didn't take very long to prove. Within 6 months, I determined it was a beta receptor. And then I went on to show that there were a variety of other hormones that regulated cyclic AMP. a variety of hormones in a variety of tissues. I worked with salivary glands, I worked with airway, smooth muscle, blood vessels, heart, liver, et cetera, et cetera. Really an exciting time. And in 1971, Earl Sutherland got the Nobel Prize for the discovery of cyclic AMP. So I spent 10 years of my education and training working with cyclic AMP. And after my residency at Mass General, I went to NIH and worked with Martha Vaughn to continue my work with cyclic AMP. And about that time, a couple of chemists had given radioactive phosphate to rabbits, isolated 2 organic phosphates in urine, cyclic AMP and cyclic GMP. That was the first discovery of cyclic GMP as a natural product. I had done several experiments with other cyclic nucleotides as a student because I found that acetylcholine could inhibit adenylate cyclase, and while many hormones would activate it in various tissues. And in my incubations, there was a sample that would inhibit the activation of phosphorylase. And I attempted to identify that molecule and worked with a variety of nucleotides, including cyclic GMP. So when it came along, I said, oh my goodness, it's another second messenger. So as I left my training at NIH, I became much more interested in cyclic GMP and worked with both cyclic AMP, cyclic GMP.
Can I ask you a question there, Fred, just for a moment?
Sure.
I think you mentioned something really intriguing to me, and that is that you had this chance to work with Earl Sutherland, who later went on to get the Nobel Prize for the work with cyclic AMP. And I was looking back at his story, and it turned out that he was the protege of Carole, who won the Nobel Prize in 1947 for glycogen metabolism.
Right.
What's his?
Carole
like...
and Gertrude Corey, his wife, were in the biochemistry, in the pharmacology department at Washington University, and they both shared the Nobel Prize in about 1947, I believe.
And it seems like this is not uncommon occurrence that there's scientific families of Nobel laureates, not by genetic relation, but by scientific association, by mentorship. This seems to be something that happens not uncommonly.
Washington University is a very impressive place, John. I think there have been about 7 Nobel laureates that came out of Washington University.
Mm-hmm. Mm-hmm. So it's in addition to
Arthur Kornberg, Paul Berg, many others.
Mm-hmm.
That were training. Ed Krebs, he had been there as well. He got the Nobel Prize later for his work with cyclic-dependent protein kinase.
So you think it's more influence of place or mentorship, or both?
I think it's the environment and certainly the mentor.
Hmm.
I remember Earl Sutherland telling us as trainees, you've got to do what's important before you're 40, otherwise it's too late.
It reminds me, I saw something written by one of your fellows recently, Dr. Scott Waldman. He said, Fred taught me that the only questions worth chasing are the big ones that can shift the mechanistic paradigm. I thought that was really interesting. So it seems as though you passed on that learning that you got from Earl Sutherland.
Well, it's really been exciting. I've been very fortunate, John, to have some outstanding mentors along the way. Mm-hmm. In college and in medical school, graduate school, and as a house officer at Mass General, and people at the NIH that I worked with as well. It was an exciting time.
I think we're probably going to get some people that want to ask.
What I often did, John, is the people are quite different, and I would try to identify their strengths and to create a hybrid that I could take a piece of this person, a piece of that, and become a different kind of individual with the assets that they all had that I could possess and use for myself.
Mm-hmm. That's a great strategy. Dr. Murad, I think people want to be joining us with questions. So I just want to remind everyone that you can join by text. If you want to ask Dr. Murad a question, you can text DEBAKEY, that's DEBAKEY, to 37607, and then text in your message, and Dr. Chanda will read your question to Dr. Murad. Or you can go online to POLLEV, that's POLLEV.com, and enter DEBAKEY. So I'm sorry, Fred, we were talking about, you had just finished your training and you were going to University of Virginia as an associate professor and to develop your independent research career. So tell us about that.
I left NIH in 1970 to go to Virginia as an associate professor in both medicine and pharmacology. I was asked to develop a new division of clinical pharmacology. And I was asked also to be director of the Clinical Research Center, which does inpatient clinical research. They had a grant which they had lost, and they wanted me to get the grant refunded. And it was a lot of effort to do that, but I took on both jobs basically and began to resurrect my own laboratory. And for several years worked with both cyclic AMP and cyclic GMP. And we learned in a few years that there were multiple isoforms of guanylate cyclase to make cyclic GMP, soluble forms and particulate forms. Different than adenylate cyclase because there's a single isoform. And with multiple isoforms, I thought there must be different compartments in the cell that make cyclic GMP, and perhaps they would have different functions, and maybe they would be regulated by different hormones or ligands. And that speculation turned out to be true. There was a soluble isoform that was activated by some of our chemicals. And in developing a guanylate cyclase assay, we put sodium azide in our buffers because of its antibacterial properties. And we can make a buffer, put it in the refrigerator, and not have to make it up every day. And with azide, it would inhibit bacterial growth. And we accidentally discovered that sodium azide activated the soluble guanylate cyclase. Not the particulate isoforms, but only the soluble isoform. My goal, gentlemen, was to find hormones that activated the soluble and the particulate isoforms. None of the hormones worked in those days, but azide did. So we put azide on cell cultures, tissue slices, a variety of tissues, including smooth muscle. One of the preparations I worked with was tracheal smooth muscle. And I learned that you don't want to correlate physiology and biochemistry in a heterogeneous population with tissue slices or organs. Because you don't know which cells are doing the biochemistry, you do know which cells are doing the physiology. And to do a clean experiment to find correlations, you need a homogeneous preparation. Blood vessels are not homogeneous, as you know. You've got nerve cells, you've got fibroblasts, you've got endothelial cells, you've got a variety of cell types. So I developed a tracheal smooth muscle. Like we would get beef tracheas from the abattoir, carefully dissect out the smooth muscle, put it in an organ bath, and we would then add agents that would cause contraction and relaxation, and monitor the activities of adenylate cyclase, guanylate cyclase, cyclic AMP, cyclic GMP. And when we put azide on these smooth muscles, it increased cyclic GMP as it did in liver slices and extracts and in heart, etc. And then I remembered, I thought it would cause contraction because I thought cyclic GMP would antagonize the effects of cyclic AMP. That was work that I had been doing with fat cells with Martha Wan at the NIH. We knew that epinephrine, glucagon, ACTH were lipolytic. They enhanced triglyceride degradation through activation of a hormone-sensitive lipase. Cyclic GMP would block that effect. It was an inhibitor. We learned that it was an inhibitor because it would activate one of the phosphodiesterases to hydrolyze cyclic AMP, lower cyclic AMP, and produce less lipolysis. So I thought cyclic G would be an antagonist. When we put it on smooth muscles, cyclic G went up, the tissue relaxed. Big surprise. Big surprise.
How did you feel when you saw that? That was not what you expected.
Yeah, it was not what I expected, but you know, most research comes with surprises, John. You think you know it all. You think you know the whole story, and you're often caught off guard. And something I learned from Ted Rall is that when you do an experiment properly with all the controls, you can trust your data.
Right.
You can believe it.
Right. Trust
and you now have to come up with an explanation to understand why it did what it did. But then I recalled as a resident taking care of patients with heart attacks in the intensive care unit. We would give them intravenous nitroprusside or nitroglycerin to decrease blood pressure, decrease afterload, and we thought we would spare the work on the heart, and the heart would heal faster as a result of that. So I suggested to one of the fellows, let's put nitroglycerin and nitroprusside on these muscles. It's got to relax. Let's see what happens to cyclic GMP.
And I guess we should say that
Fair enough, it increased cyclic GMP.
And we should say at this point, no one knew how nitroglycerin worked. We've been using it for 100 years.
Nitroglycerin was discovered in 1847 by Italian chemists. What I've learned what chemists often do when they make a new compound is lick their finger, stick it in the powder, and taste it. And when they did that, they got vascular headaches. Mm-hmm. And the physicians in Europe realized that nitroglycerin was a vasodilator. And in the 1870s and 80s, they started using it for angina pectoris in patients with heart attacks.
In fact, Alfred Nobel took nitroglycerin for his angina.
Exactly. Alfred Nobel made his fortune taking nitroglycerin to make dynamite. That's how he made his fortune. And when he was prescribed nitroglycerin for his chest pain, he also had a stroke at one point. He refused to take it because of the vascular headaches. He didn't want to take it.
So can you, just for the people listening, can you tell us how it is that we can use nitroglycerin for cardiovascular health? And on the other hand, it can also be used to make dynamite. Tell us a little bit about that.
Well, it's a trinitro. It's glycerol with 3 nitrates on it. It's explosive. Mm-hmm. So you have to be very careful with it. If you have the liquid, you can't shake it and vibrate it, it will explode. But what you can do is to convert it to nitric oxide, and that's what we learned. It was a prodrug precursor converted to nitric oxide, and that requires an enzyme to do that. Mm-hmm. Nitroprusside, however, spontaneously gets converted to cyanide and nitric oxide in the bloodstream. You don't need an enzyme to convert nitroprusside to nitric oxide. Mm-hmm. So we ended up testing a lot of nitrogen-containing compounds that were all vasodilators that increased cyclic GMP. They activated the soluble form of guanylate cyclase. Mm-hmm. And with all these different structures, I said, my goodness, they can't be working the same way. They got to be working through some common factor. What is it? And it was quite a mystery story for a couple of years as to what they were being converted to. Mm-hmm. And fortunately, along the way, we learned that in some tissues, nitroglycerin and azide would not work because there was hemoglobin in the preparation, and hemoglobin scavenges nitric oxide. We also added methylene blue as an oxidant, and it blocked the effects of azide as well. So we said, are all of these compounds, nitrovasodilators, we coined the term, converted to nitric oxide? And we then one day, in early December 1976, made nitric oxide in the fume hood by mixing sodium nitrite with sulfuric acid and ferrous sulfate, and it created nitric oxide gas that we vented into our incubations, and it activated all the soluble form isoforms of guanylate cyclase from all the tissues we tested.
That was the Eureka by the way. I said that sounds dangerous, by the way.
Activated an enzyme.
That sounds dangerous, by the way. And
well, you have to be careful.
you were telling me that when you were a kid, you anesthetized butterflies. That sounded
right.
Pretty dangerous too. You're 10 years old. You went. Tell everyone. You went to the chemist. You went to the pharmacy.
I used to living on Lake Michigan, and I would go to the marshes and catch butterflies. I wanted to mount them on a piece of cardboard in my bedroom, but I didn't want to grab their wings to mount them and damage the color of their wings. So I went to the local pharmacist and I said, "Can I have some potassium cyanide and hydrochloric acid?" And he gave it to me. I went home, put potassium cyanide in a jar, a mason jar, punched a hole in the lid. And when I catch a butterfly, I transfer it from the net to the jar, put on the lid, and then carefully drop some acid through the hole in the lid to create cyanide gas and put the butterflies to sleep. Now I can mount them without their flapping all their wings.
Did your parents know that you were doing this?
No. No. If I were to do that today, I'd probably be arrested as a terrorist.
Yeah. Well, it gets back to this risk taking. You are a risk taker, aren't you? In science.
Yes. I think you have to take risks to get ahead. Yes. Mm-hmm. Sometimes you make mistakes, but sometimes you don't. Mm-hmm.
Well, you didn't make many mistakes, and you were just telling us a story how you discovered that the nitric oxide was the mediator of all these effects of the nitrovasodilators, and that was amazing. This gas was the mediator of these effects of nitrovasodilators. That must have been stunning when it occurred to you that this simple molecule, nitric oxide, was having all of these effects.
It was unheard of that a gas in a free radical could activate an enzyme. And I thought that was really important, interesting, novel chemistry, and we got to pursue it and figure this out. How does it do it? It turns out that the soluble isoform of guanylate cyclase is a heterodimer. On the beta subunit is the heme prosthetic group. The heme has a ferrous iron. Nitric oxide has a high affinity for transition metals, particularly iron. When it binds to the iron in the heme lifts off the enzyme and removes the inhibition of the enzyme. The enzyme's activated a couple hundredfold to make cyclic GMP from GTP.
Beautiful.
But that took a lot more work in chemistry to figure that out.
That's beautiful and well deserving of the Nobel Prize. Well, tell us now about the clinical implications of this work. You've shown that nitric oxide is a vasodilator, and subsequently, maybe tell us a little bit about the discovery that led you and the other Nobel laureates to the idea that this was an endogenous molecule, that the blood vessels could actually make nitric oxide. How did that come about?
That really got started with the work of Bob Furchtgott. Robert Furchtgott was the chairman of pharmacology at SUNY Dowstate in Brooklyn. And I had met him as a student. Earl Sutherland would often invite prominent pharmacologists, biologists to come and give lectures. And I had dinner with Furchtgott, so I got to know him a little bit. And he was invited to give a seminar at the University of Virginia in the pharmacology department around 1980, 81. Early, I think it's late 1980. And he talked about endothelial-derived relaxant factors. It was known that drugs such as acetylcholine, thrombin, acetylcholine, ATP, would cause relaxation of blood vessels in intact animals to lower blood pressure. They lowered blood pressure. But if you put them on blood vessels, they had no effect. And that's because when you handle blood vessels to dissect out the smooth muscle, you destroy the integrity of the endothelium. And Bob one day asked his technician to make some segments, and instead of making longitudinal strips where you would handle the endothelium and destroy it, it's very fragile, he decided to make sausages, like chopping off rings, and found that acetylcholine caused relaxation. And Bob's pretty clever. He realized the difference between rings and longitudinal segments was the presence of the endothelium. So he did his famous sandwich experiment. He took a vascular strip with endothelium and one without, and he strung them up in an organ bath adjacent to one another. And what he put in, endothelial-dependent vasodilators such as acetylcholine, both strips relaxed.
So something was
coming from the
diffusing.
endothelium that caused relaxation of both muscle segments.
Yeah, something was
He
relieving the
called that
endothelium.
substance endothelial-derived relaxant factor. He gave that seminar in Virginia while I was still there. Endothelial-derived relaxant factor had a short half-life, only a few seconds. And it was also blocked by agents that we were using as inhibitors of guanylate cyclase activation, hemoglobin and methylene blue. And after his seminar, I said, Bob, come to my office. I think EDRF is going to be a reactive species, perhaps nitric oxide, and it's got to work through cyclic GMP, calcium, or cyclic AMP. Let's figure this out. We agreed to collaborate. During that visit, my wife took his wife to Monticello to see the cast of the home of Thomas Jefferson. She tripped on a step, broke her leg. The x-ray revealed metastatic breast cancer. Bob then returned to New York to get her treated. The collaboration never took place. He was going to make samples, send us frozen extracts, and we would assay for cyclic nucleotides and cyclase activities. That never happened. Mike Peach, a professor at the pharmacology department at Virginia, was also interested in vascular biology. He was looking at the effects of prostaglandins through cyclic EMP production to relax vascular segments. And he was called to Syntex as a consultant, and my lab and office was across the street at the VA hospital. And afterwards he came to my office and he said, "Fred, what are you doing with EDRF?" I said, "We're waiting for samples from Furchgott. He's supposed to send them, but he hasn't." I said, "Let me call one of the fellows in the lab who was supposed to get the samples." Robert Rappaport. Robert came to my office. I said, "Robert, have you ever gotten samples from Furchgott?" He said, "No." But then he sort of smiled. And I said, "Robert, what's going on?" He said, "I've done the experiment. The samples are in the RefCo, but I haven't assayed them." I said, "Robert, you're going to assay them tonight, and tomorrow we're going to know if they work through cyclin G." And sure enough, they did.
Wow, that's a great story. So something was being released from the endothelium that was stimulating cyclic GMP production, and it had to be nitric oxide or something similar to nitric oxide based on your work.
Right.
Yeah.
Since it was blocked by hemoglobin and ethylene blue, I thought it was nitric oxide or some complex of nitric oxide.
That's great. And so the
in fact, in one of my reviews, I think for JCI, I wrote a review and I called EDRF the endogenous nitrovasodilator.
So fast forward now that we've learned that endothelium-derived nitric oxide relaxes blood vessels, what have been the clinical implications of it? What does it mean to the physician? What does it mean to the layperson that's concerned about their cardiovascular health?
Many patients have what we call, John, as you know, endothelial dysfunction. Their blood vessels don't make enough nitric oxide. And they don't make enough for a variety of reasons. One of them is because of the compound that you discovered, asymmetric dimethylarginine. Arginine can be converted as a substrate to nitric oxide by an enzyme, nitric oxide synthase. It has cofactors that have to be in the reduced state to be effective. If you oxidize those cofactors, such as tetrahydrobiopterin to dihydro, it no longer makes NO, it makes superoxide, which will scavenge the NO to make a toxic molecule, peroxynitrite. So by understanding that these blood vessels from patients with hypertension, diabetes, atherosclerosis, tobacco use, and maybe obesity don't make enough nitric oxide, we and others have worked out the biochemistry to figure out why, and we can figure out ways now to treat these patients. It also tells us, John, that when you have these patients with heart attacks, they're likely to have endothelial dysfunction. So they're not going to respond to the classic vasodilators. In order to lower blood pressure, decrease afterload, you have to use direct-acting nitrovasodilators that work on the smooth muscle. So that influences how you manage blood pressure in the coronary care unit.
Now, there are other things that nitric oxide can do in addition to relaxing blood vessels. It also has effects to preserve vascular homeostasis. Tell us a little bit about that, so the other effects of nitric oxide to prevent vascular disease.
John, there are today 170,000 research papers on nitric oxide, and there are very few things that it doesn't do. It's incredible.
It is.
It's really incredible.
I was referring.
It inhibits platelet aggregation and blood clotting. It regulates cell proliferation. It influences differentiation and proliferation of embryonic stem cells. It influences cancer angiogenesis to feed the tumors. There are all sorts of interesting applications. And for the past 15 or 20 years, we've been focusing a lot on stem cells and cancer with nitric oxide and cyclin G.
Yeah. We'll get to that in just a minute, and just to for the clinicians and the sophisticated lay folks that are following, what can we do to improve our nitric oxide production? It seems like it's a very important molecule to preserve vascular homeostasis and prevent vascular disease. What kinds of things could we do to improve the release of nitric oxide?
Well, nitrates and nitrites are converted to nitric oxide. They're precursors. They can be converted to be reduced to NO. So foods, leafy vegetables, spinach, kale, other things have a lot of nitrite and nitrate. It's a substrate to make NO. There are nutritional supplements that have nitrite and nitrate in them. There are nutritional supplements that have L-arginine and various antioxidants to keep the cofactors reduced for NOS, for the nitric oxide synthase. So exercise enhances the expression of the endothelial form of nitric oxide synthase. There are a variety of approaches.
And there's also medications.
Exercise, diet, nutrition, nutritional supplements.
And what about medications? What medications can enhance or protect the endothelium and preserve nitric oxide? Statins, for example, I think can increase NO synthase expression and increase nitric oxide generation.
There are some interactions of nitric oxide in the statin pathway, yes.
Yeah. Well, great.
I haven't worked with those pathways. My colleague, Mike Brown and Joe Goldstein were my interns as a resident. They're probably the people to talk to about that.
Right. You mentioned that you were studying now nitric oxide and its role in stem cell biology. Tell us a little bit about that.
Right. We have found that embryonic stem cells from mouse and human can differentiate. And if you manipulate the nitric oxide cyclic GMP production in those cells, they proliferate faster, and they can differentiate into certain niches. We can make them become neuronal cells or myocardial cells. How it's doing it, I don't know. We know that NO does regulate gene expression. How it's doing it is difficult to figure out. It's probably an epigenetic mechanism on the chromatin complex. For example, in tumors, most cancers that we've looked at, glioblastoma, lung, breast, pancreas, prostate, have an abnormal soluble guanylate cyclase. I told you that soluble cyclase is a heterodimer with alpha and beta. What happens in cancers is they lose alpha. They only have beta. They cannot respond to nitric oxide. To make nitric oxide by activating soluble cyclase, you need the heterodimer with the prosthetic group E. Cancer cells have an abnormal soluble cyclase. But what happens, the beta subunit migrates to the nucleus and comes down with the chromatin complex. It's somehow regulating gene expression.
Interesting.
And we'd love to figure out how it's doing it, because it could offer us some novel approaches for cancer therapy.
Just on that point.
We do know that most cancers have high levels of NOS2, the inducible form of nitric oxide synthase. I think they make a lot of NO because it has angiogenic effects to proliferate endothelial cells and block the proliferation of smooth muscle cells. So you make microvessels to nourish the tumor.
Right.
If the tumor had soluble cyclase where NO could activate it, the cyclic GMP as a tumor suppressor, it would kill itself. So it suppresses the expression of alpha, makes a lot of NO to make blood vessels so it's nourished to survive. So now there's several ways to approach cancer therapy, by influencing angiogenesis or by influencing the cancer, because NO has effects on angiogenesis and cyclic G. Cyclic G is tumoricidal. It binds to something in that chromatin complex to influence gene expression and the survival of the tumor. I'd like to know what it is. Speaking
of the nitric oxide pathway and epigenetic effects, actually, Dr. Chanda has one slide that we should show. Why don't we pull up the slide, fellas? Why don't we pull up that slide that we gave you and get Dr. Murad's comment on a new epigenetic modification by nitric oxide. You want to take us through this slide?
Sure.
Polish?
Dr. Murad, it's a pleasure for me to speak in front of you and represent our lab work in front of you. So this slide represents the story of transflammation that our lab has been doing since our paper was published in Cell in 2012. It summarizes the transflammation and the role of nitric oxide as an epigenetic modifier. So the transflammation process starts whenever a cell gets assault in the form of a pathogen. It's a bacterial pathogen or virus, or it might be a cell damage. And cells have sensors in the form of PRRs, like pathogen recognition receptors. They could sense these PAMs, the pathogen-associated molecular patterns, or damage-associated molecular patterns. And like these PRRs, like TLR3 or RIG1, they get stimulated, and in turn they activates the innate immunity. Activation of innate immunity leads to expression of the cytokines, and this story is known, this story is well characterized, but what we discovered is another limb of the activation of innate immunity. What we discovered is like activation of innate immunity also leads to a global change in the expression level of the epigenetic modifiers. Histone deacetylase level goes down and histone acetyltransferase level goes up. Histone deacetylase removes the acetyl mark from the histone protein, leading to decreased DNA accessibility, whereas histone acetyltransferase transfers the acetyl mark to the histone proteins and it opens up the chromatin, means more DNA accessibility. So here you see that in presence of poly-IC, which is a TLR3 agonist, it stimulates TLR3, and then it leads to the more expression of NF-kappa B. Now, in NF-kappa B activates more expression of iNOS, which then translocates to the nucleus, and it nitrosylates epigenetic modifiers. In our study, we found at least 2 epigenetic modifiers. One is the MTA3, shown over here. MTA3 is a subunit of NURD complex or nucleosome remodeling complex. It removes the acetyl from the chromatin and closes the chromatin. When MTA3 is nitrosylated, its activity is inhibited and the chromatin opens up because this complex falls off from the chromatin. Similarly, during transdifferentiation, we found that ring 1A, which is a subunit of polycomb repressive complex, it also inhibits opening up of chromatin. So both these nitrosylation events by nitric oxide helps the repressive complex to fall off the chromatin and opens up the chromatin, and this leads the cell to a state, a plasticity, a fluidity, depending on the transcriptional direction available to these cells. It can go either towards pluripotency, or if any other lineage factor is present like endothelial cells, it can choose its direction to go.
So what do you think about that, Dr. Murad, about nitric oxide as an epigenetic modifier?
Well, transcription is pretty complicated. There's so many players, that's really making it difficult. We're trying to figure it out as well. When you activate NF-kappa B, and there are many ways to do that with IL-1, interferon gamma, TNF-alpha, you activate NF-kappa B, that enhances DOS2 production, make a lot of NO. NF-kappa B also enhances the expression of a lot of pro-inflammatory genes as well. So you're making other free radicals, and you're probably going to make peroxynitrite for the nitrosylation reaction. And what players are getting modified by the nitrosylation? I suspect there are lots. Now, Jonathan Stamler tells me there are more than 100 proteins that get nitrosylated by modifying the hydroxyl adjacent to the group on tyrosine adjacent to the hydroxyl, or by modifying the thiol groups. They're reactive and they're all talking to each other. And I think the question is, what is the rate limiting reaction here that's a target to come up with novel therapy for cancer or stem cell biology? That's going to be key.
Yeah, I think that's right. And there's basically, we think of this as a way in which the cell, when it's challenged by a pathogen or by damage, it's a signaling pathway that basically opens up the chromatin, increases DNA accessibility so the cell can reach back into its genetic toolbox and pull out whatever it needs to survive or adapt. In the case of cancer, we do think that this pathway may be activated, or at least activated during some point in the evolution of cancer, increasing DNA accessibility. So inflammatory signaling has this very interesting effect to increase DNA accessibility and increase cell plasticity.
Right. Now, as you activate inflammatory genes and make these reactive species, you're going to modify NO.
Yeah.
You're going to modify the production of NO by inducible NOS. You're going to also, by oxidizing the cofactors, but you're also going to form complexes with what NO is being produced by the other isoforms.
We had
Or bro-
we just got a question from the audience, and I think you could talk a little bit about this. How does nitric oxide transmitted from NO synthase? How is it transmitted from NO synthase to guanyl cyclase? Are there molecular transporters for NO?
No, no. When it comes out of cell, it binds to glutathione. Glutathione is a transporter for NO. So NO is really a hormone carried to distant sites by the glutathione.
So this is really an interesting idea, isn't it? I mean, NO is a free radical by itself. It's not going to last very long. It's going to get destroyed in the cell. But if it binds to something like a thiol group and creates a nitrosothiol, then it can persist for longer periods of time.
Right. In fact, there's an enzyme that reduces glutathione and prevents it from carrying NO. And one of the companies I advise has come up with compounds that inhibit the glutathione reductase to permit the delivery of more nitric oxide. And in a variety of animal model experiments, they found all sorts of interesting nitric oxide effects. Mm-hmm. So that drug might be very useful for all sorts of clinical problems. But when you get involved with free radical, that's why discovering it was so exciting to me, John. A free radical, an oxidizing species, an unusual molecule, a gas that does something interesting in biology. Mm-hmm. And now the list has grown and grown. Simple molecule with so many complex activities. You talked a little bit about serendipity. Let's talk about that for a moment, about that, the happy circumstance. When you gave azide, when you found that azide activated guanylate cyclase, you didn't expect that. Other examples of serendipity are prominent in the literature. Tell us, what is the role of serendipity in scientific discovery, in advancing an idea, in advancing a career? Tell us about what role serendipity plays. Well, biology or biochemistry is pretty complex. Mm-hmm. And we can't predict everything. We think we know something, we don't always know. Mm-hmm. And I think we have to be prepared for surprises. Mm-hmm. And that's something I learned from Ted Rall. Ted used to insist that I have all these controls in my experiments. The experiments were so big, I used to call them Sunday experiments, mm-hmm. because Sunday was the only day I had a lot of time to do a big experiment with all of Ted Rall's controls. Mm-hmm. But if you did the experiment properly, you could believe your data. And that's something I learned from Ted. If it's well done, well controlled, you can believe what you see. And it may be a surprise, but you've got to believe it. That's interesting, because we've kind of evolved to the same approach. I tell my postdocs that if you do the experiment correctly, the data tells you the truth, and you have to follow that truth. And even if it is against your hypothesis, maybe it's not what you wanted to see, but it can lead to something even more important than what you were originally thinking of. I agree. Tell us what you think the next big thing is in nitric oxide, in this field of nitric oxide and endothelial health. Well, I think there are going to be a couple of things, John. I think we're going to figure out a way to use this information for cancer therapy. And it may be by altering its ability for promoting angiogenesis. Or maybe it's going to be a way to get that target in the genomic complex regulated, nitrosylated. Mm-hmm. And maybe in stem cells, it'll permit us to make the cells we want for various transplantation studies in biology. Mm-hmm. We know now that you can grow stem cells on various surfaces. Will there be the day that we can make artificial valves and hearts and whatever in the laboratory? I think that's going to come in another 20 or 30 years. Mm-hmm. And I think by understanding these messengers and how they influence that process, proliferation, differentiation, could be very useful tools. Mm-hmm. We only have a few more minutes left, but we've all been dealing with this pandemic, and we're starting to get to the other side of it. But a new pandemic seems to be arising. This long COVID or right. Prolonged symptoms related to a COVID illness.
a lot of confusing symptoms in this long COVID disease. What do you think the role might be of endothelial dysfunction in the manifestations of this disease? The fatigue, the impaired cognition, the microvascular thromboses that occur, blood clots that occur in these patients. What role do you think of an endotheliopathy may be playing in this condition?
Well, that's a good question. You know, we used to think years ago that the endothelium was sort of a benign layer. The only function was permitting agents to go from blood vessels to the underlying tissues. We now know that it's a very active cell type, a huge cell type. You tally up all the endothelial cells, it's an organ probably bigger than the lungs, the heart, the liver, the brain. And it has a lot of mediators that regulate it, and it responds to a lot of different agents. And it's a very important system.
So, in other words, an endotheliopathy could have a lot of different side effects.
I've advised a number of companies dealing with nitric oxide over the years, mostly nutritional companies. But there are two companies that are using nitric oxide to treat COVID virus. One has an inhalation product that we're using for sinusitis. And I said, since it's antibacterial, antifungal, antiviral, why don't you see if it's going to kill the coronavirus with a nasal spray? And they now are in the midst of several clinical trials in England, Canada, in Israel, where in fact it does work. It kills the coronavirus within 30 seconds or 60 seconds as you spray it in the nose. Another company is taking atmosphere and converting nitrogen in the atmosphere through an electronic device to make plasma, a gas. And they spritz the gas on diabetic ulcers and they heal much more rapidly. We know that topical NO in mice and rats, if you do burns or punch biopsies, the wounds heal in about half the time. It has a variety of effects. It has angiogenic effects. It recruits stem cells. It enhances the epithelial cells to close faster, to close the wound. It has antibacterial, antiviral. It's got a lot of other effects. So it's a pretty common messenger that came along way back. It's a simple messenger. And now it has very complicated biochemistry and biology that we're trying to sort out.
It's an amazing story. And Dr. Murad, thank you so much for sharing it with us today. And congratulations on a spectacular career and making transformational changes in how we deal with our patients. Thank you so much for that and the time you spent with us today.
I think we should be talking more about epigenetic regulation. Perhaps we can collaborate in some way.
That would be fun. We'd love to collaborate with you.
We know that cyclic G and NO bind to that complex. It comes down with polymerase II, and it does regulate the transcription of P53. So it has a lot of effects.
Well, let's take a look.
Yeah.
Love to do that with you. Thank you,
All right,
Dr. Murad.
thanks, Scott. Been fun.
Bye-bye.
All right.